see you. You can see you. Yes.
Okay. So it's my pleasure to welcome Professor Dr. Soumitra Das to deliver this lecture today on hepatitis C virus, life in the liver. So as you know that this is a topic which is also of primary interest to me, being a gastroenterologist. And hepatitis C, as you know, is a disease which affects millions of people worldwide and is the cause of end-stage liver disease called cirrhosis of the liver, and also ultimately results in hepatocellular carcinoma. It leads to much morbidity and mortality. And there is no effective vaccine. And until recently, there wasn't any effective treatment either. So it's a wonderful thing to listen today to Dr. Das for giving this, and who will give this lecture on hepatitis C virus. Let me also introduce him to the audience here. Dr. Das obtained his PhD from University of Calcutta in 1992 at the Indian Institute of Chemical Biology, and studied initially Leishmania donomini, which is also one of the important pathogens which causes the dreaded disease Kala-Azar, and of course it is quite prevalent in India. And then he worked as a postdoctoral fellow at UCLA, one of the most prestigious universities in the world, and subsequently has been working on various viruses, including RNA viruses, hepatitis C, Coxsackie, dengue, and now currently SARS-CoV-2. And his focus of his lab has been now to develop effective molecules against hepatitis C virus and also develop new vaccines. I think the vaccine also against SARS-CoV-2 he has been involved in developing. And I have come to know him only last 1 year or so, because he was heading the INSA-COG consortium, which is Department of Biotechnology platform to have nationwide genomic surveillance of SARS-CoV-2 and emerging variants. And I have seen him firsthand, his leadership, how he has handled a diverse group of people. In every meeting, there are 30, 40, 50 people, everybody having, divergent opinion, and everybody wants to do this and that. And in a very calm voice, he conducted those meetings. And I really appreciate that, Soumitra, when you had as the convener and chairperson of those INSA-COG meetings. And also thank you very much for including THSTI as one of the INSA-COG partners. In fact, he said right in the beginning, oh, that's not a problem. THSTI, of course, will be a partner in that. And ever since then, that THSTI has been sequencing the virus here. So over to you, Soumitra, and maybe Ramandeep, if you have anything else to add or say before he takes over the podium and start his lecture.
So we can start the lecture.
Okay.
Welcome.
Over to you.
Dr. Das, you can start the lecture.
All right. So, I mean, it's a great pleasure. I just wish if I could go there physically and then interact with you, because, nothing like that, giving a lecture in front of the audience rather than in this virtual mode. But as I was just telling some of you that I just moved back to IISc and then settling down at IISc after 3.5 years. So I thought since it's getting delayed, actually Milan has been asking me from December. So I thought let's uncouple it and then give the lecture. And then I will visit definitely in some other occasions, THSTI. But it's always a pleasure to interact all of you. And I had several occasions in the past, and I really enjoy that. And as I was referring that THSTI has done played a tremendous role in this COVID research in last 2 years. And we are proud of that. And we were just discussing that when several laboratories were actually closed down across India, some of those laboratories, they couldn't really afford to close down, and they just really stood out to the promise and then actually helped the nation. So kudos to all the leaders and the team of THSTI. So today I'm going to talk about hepatitis C virus, as Pramod rightly mentioned, that my lab have been working almost 23 years at the Indian Institute of Science on this hepatitis C virus, although we do work on other viruses, Coxsackie B3, which is something similar to poliovirus. We say it is a close cousin. We also work on dengue virus, and we started thinking on SARS-CoV-2. But hepatitis C virus is the central project that we have been working on. Now, if all of you can switch off your microphone. So hepatitis, as you know, can be actually contributed because of alcohol, because of autoimmune disorders, could be non-alcoholic steatohepatitis, could be toxins or other infections, and mostly it is also done by the viral hepatitis. Now, when we talk about the viral hepatitis, there is a range of hepatitis viruses with all the alphabets A, B, C, D, E. Only F, I call it, as actually failed to sustain. So we don't have HFV, but we have HGV. And you can see there are, all of them are single-stranded except the HBV, which is actually a double-stranded DNA virus. And, all of them actually targets the liver. And that's how I thought that how they actually crosstalk in the liver, how is their life. We tried to understand over this almost last 25 years. I started this hepatitis C research when I was in UCLA. So we're trying to understand what is the crosstalk between the host and the virus. As there are several literature available that hepatitis A, there are proteins identified which actually causes pathogenesis. And hepatitis E, I'm sure you people are aware of it because Milan has been heavily contributing into this hepatitis E research at THSTI, WARF1, WARF3. Both of them have been shown to be contributing to pathogenesis. Hepatitis Delta virus, the S antigen, L antigen, both of them. And then for the hepatitis B, HBX, and for hepatitis C virus is actually 4. So many a times it actually contributes directly to cirrhosis or it causes a cytopathic effect. Sometimes it contributes to the immune response. And in a couple of cases, it actually goes to hepatocellular carcinoma. But today's talk, I'll be only restricting to hepatitis C virus, as I told you in the beginning, which is identified in 1989. And it's known to cause liver cirrhosis that often leads to hepatocellular carcinoma. As Pramod was rightly mentioning that, it's actually, it's a dangerous disease in the sense people doesn't really notice that they are harboring the virus for a long time. And by the time, if it is undiagnosed, 75 to 85% cases establishes chronic hepatitis. And of that, 20 to 30% develop cirrhosis. And of that, 2 to 7% actually ends up with the end-stage liver disease or leads to hepatocellular carcinoma. Now, if you look at the virus, it's a positive strand RNA virus. So the positive strand RNA virus initial obligatory step is to make the viral proteins. So the viral proteins are made, and then the viral proteins helps to make from positive strand to the other strand, which is called negative strand. And the negative strand is then actually relocates into the membranous web structure used as a template to make more and more positive strand. Now, viral proteins helps in replication. The virus also hijacks some of the host protein to help their replication. And that's the subject matter today. I'm going to decipher the mechanism how this translation and replication happens in hepatitis C virus. So, and then the positive strand, which are made in much abundance, actually much more, sometimes 10 to 20 times or sometimes 50 times more than the negative strand RNA. And these positive strand RNAs are packaged and then finally egresses out and infect the neighboring cells. Now, all these things happens in the liver. And if you really look at the liver, it has hepatocytes, which are 80%, endothelial cells, there are stellate cells, and there are Kupffer cells. So, for me, it was really surprising at the time to understand how come the virus is so specific that it infects hepatocytes and really doesn't really, at that time, the knowledge was that it doesn't really affect the stellate cells or the Kupffer cells, the neighboring liver cells. So almost imagining a situation that the virus knocks at the door, or they have the key because of the receptor ligand interaction, lock and key, opens, gets into the room, and then goes straight to the kitchen. They exactly know where to go. Go to the kitchen and then use all these vessels and utensils to make palatable food. And this host actually remains outside the kitchen, and most of the time they starve to death. Now, the palatable food is really palatable. It looks like almost a big sub sandwich, and that sub sandwich actually is cut it into structural and the non-structural protein. And this cleavage is done by the host cell signal peptidases and some viral proteases like NS3 or NS2, NS3 cleavage sites are there. And this structural protein and non-structural protein are mostly the functional, and they are being targeted for designing antivirals, because without that, virus cannot replicate. As you all know, that initial years, soon as hepatitis C was actually being noticed, it was treated with pegylated interferon and ribavirin. But 40% cases were actually successful, but most of the cases it was not successful. Until recently, when we have some protease inhibitor like telaprevir, or the polymerase, RNA-dependent RNA polymerase, which is NS5B, sofosbuvir is actually inhibits to that polymerase, and this actually hits the market heavily, and it actually helps to cure most of the cases. Although there are certain cases where there are issues with the emergence of drug resistance mutants or the relapse once the drug is stopped. So, people keep on looking for additional or alternative therapy approaches to circumvent this situation. There are also, keep on the drugs. I'm sure Pramod will have much more updated information about those drugs that are actually upcoming to target different enzymes of the hepatitis C virus. But what we were actually interested at that time when I started this lab at UCL, at the Indian Institute of Science, that how we make proteins and how the virus make their proteins. Is it different? Because there are actually some instances, it has been shown that the virus synthesizes their protein in a completely different mechanism. So we try to initially understand how we make our proteins and how differently the virus make their proteins. So how we make our proteins, as we know all from the text, that mRNAs are capped at the 5' end, which is 7-methyl-guanosine cap. There are cap binding protein, which is eukaryotic initiation factor that binds and recognizes. Then EIF4A, which is a helicase, joins. Then EIF4G joins. And EIF4G, I call it like a grandfather who holds the whole family together. And eventually, the EIF2 and EIF3 brings in the 40S, scans for the initiator AUG, and then the 60S joins and then starts transmission. This we have read in the text, and it's been going for a long, long time. But if you look at the hepatitis C virus RNA, it doesn't have a cap at the 5' end. Instead, they have a structure which we call an RNA scaffold that actually attracts the 40S to come directly bind onto that site. This was actually originally named as ribosome landing pad, and later on it has been rephrased as internal ribosome entry site, or IRES. Now this IRES is closer to the initiator AUG. Virus does not have to bind to the end of the RNA and traverse all the path to look for the initiator AUG, but it can bind internally to a sequence closer to the initiator AUG and immediately find out the 60S to join, and then the translation starts. Now, a couple of things to remember. First of all, the viral RNA is uncapped, and the 40S binding to the RNA does not need eukaryotic initiation factors like the other viruses, like even for poliovirus, it needs some eukaryotic initiation factor. But for hepatitis C, the 40S binding is almost like a prokaryotic type, where the 30S directly can go and bind to the Strand sequence. So here 40S can directly bind to the IRS. And when we started the lab, the similar time, around 2000, the year Jennifer Doudna, who actually got the Nobel last year, along with Charlie, for the cryo-electron microscopy, and for instance, Charlie Rice also got it for hepatitis C virus work. Now, what Doudna showed that this is a cryo-electron microscopy picture of the hepatitis C viral RNA along with the 40S subunit. And she demonstrated that viral RNA actually binds to the exit gate of the 40S ribosome, triggers a conformational alteration, and access to the front gate where you have the the A sites and the P sites. So by doing so, what they do is they achieve to avoid the competition at the front gate. The front gate, remember, the cellular mRNAs are all lined up to get the 40S binding to the A sites and the P sites. So the virus has tactfully go to the go to the exit gate, somehow bribe the gateman, and then gets into the 40S access, and then make use of the front gate to have the translation going. So since it is unique and fundamentally different, I was very much fascinated to find out, can we really understand the mechanism, find out what are the ribosomal proteins that are involved for this viral RNA to bind to the ribosome, what are the host factors that actually helps or the virus exploits for this activity, and what are the viral proteins that are actually involved in the regulation of the translation. So, and the idea is, remember, as rightly pointed out by Pramod, that still there is no vaccine. And when I started this around 1998 to 2000, there was no treatment or no drugs available. So obvious question was, can it be targeted to design a selective antiviral? Because it's a unique and fundamentally different mechanism, we hope that it won't really interfere. Actually, we demonstrated that if you really take a stem loop structure of the 5' untranslated region, different stem loop structure, there are 4 such stem loop structure, 1, 2, 3, 4, and we demonstrated that if you take a sub stem loop structure, use that as a competitor, then this structure can go and then sequester the same place where the viral RNA actually interacts with the 40S ribosome. And it prevents the viral RNA interaction with the 40S, means it inhibits the viral RNA translation, but does not really affect the front gate of the mRNA attachment with the 40S. So this is first time we demonstrated that ribosome and HCV RNA interaction can be a target for designing antivirals. And that's how we have patented, and we are actually trying to commercialize that whole thing. But it's not so simple. Over the years, I have learned quite a lot. And we found that this IRES actually, first there is a RPS5, a ribosomal protein S5, which actually demonstrated that this small RNA that was able to inhibit the viral RNA translation actually goes and binds to the ribosomal protein S5. This S5 protein actually sits in the exit gate, and that's the protein the virus was bribing to get into the exit gate and then get entry into the ribosome. Now, since this RPS5, then we found not only the RPS5, it needs some host factor. This host factor, largely we have worked is the LA protein, which is human LA autoantigen. The LA is actually found in the lupus disease, lupus erythematosus, and this is a nuclear protein. It has nuclear function, it helps in tRNA maturation, but somehow in the virus-infected cells, virus drives this protein out, and this LA protein needs to bind near the IRES, unfolds the structure, and helps the 40S to bind on to translate. So that means alone RPS5 is not sufficient. You need the LA protein to be in the near vicinity so that the translation can kick off. And we also eventually showed that there is another protein called a polypyrimidine track binding protein, or PTB, which is again a factor needed for alternative splicing. Nuclear protein comes out to the cytoplasm, helps the virus to translate. And, by the time, it was a co-evolving story where Peter Sarnow's lab from Stanford showed that MIR122, it's a microRNA, first time demonstrated that it binds to 5' UTR rather than binding to 3' UTR, because at that time knowledge was microRNAs are actually designed to bind only to the 3' UTR, not to the 5' UTR. But he showed that microRNA 122 is very unique in the sense it binds to the 5' UTR. Not only that, it helps the RPS5 or the ribosome, 40S ribosome, to bind onto the viral RNA and helps in the translation assembly. First 10 years is gone. Then we just tried to find out, it looks like a huge orchestra where there are LA protein, there are PTB protein, there is another protein. One of my students, Shweta, who is now working in Peter Sarnow's lab in Stanford, she showed that there's a protein called HUR, which binds to 3' UTR and actually helps the virus to initiate replication. In between, we also showed that LA protein helps to translate, But there is a viral protein called NS3 protease that also has some RNA binding ability. The NS3 protease goes and binds to the same place where the LA binds, kicks out the LA binding from the 5' UTR and stops transmission. Why? Remember, the viral RNA is positive strand RNA. The same RNA will be used for making the protein, and the same RNA has to be used for making a replication. So the RNA used as a template for translation and template for replication. So unless there is a switch between translation to replication, there will be a head-on collision between the advancing translational complex and the reversely advancing the replication complex. So what we demonstrated that actually the viral protein, when it actually reaches to a quantum amount or quorum, then that particular protein NS3 goes and kicks out LA. And this LA now comes out and binds to the 3' UTR. So two things we demonstrated, that these are nuclear protein LA PTB. They come out from nucleus to the cytoplasm. Then they bind to the 5' UTR to help in translation. They also binds to the 3' UTR to regulate the replication of the virus. And somehow we found this is, there are more complexity by regulations by microRNA. MIR122 is one, but we have also shown there is another microRNA 125B, which actually regulates HUR protein, and that's how its abundance is regulated. And HUR binding is central to initiate the replication. So looks like there are enough character to have a real orchestra with the proteins and microRNA, but how about the long non-coding RNA? So that's the time when Gitika started looking at it. That we tried to have the schematic and show that we are working on the proteins like PTB, LA, HUR. We demonstrated microRNAs, MIR122, MIR125B, and MIR375 I'm going to talk about today. And there is long non-coding RNA. Can we really find out what are those long non-coding RNA? And eventually we showed there are 3 long non-coding RNA which actually plays major role in the virus replication and pathogenesis, ALK, MALAT1, and NEAT1. And ultimately, all of them leads to the hepatocellular carcinoma. And very recently, we started looking at it from a different perspective of P53, and it's one of the isoform which lacks the first 40 amino acid. Our lab have been working long time with that, and we first demonstrated this is generated by an IRES-mediated translation of P53, which makes a truncated form. And this P53 and delta 40 P53 decides who is going to actually pronounce the effect on the hepatocellular carcinoma. And that's the thing we are going to talk about some other time. So what we all talk so far is that the virus binds to the membrane, and there is a signal from nucleus, the proteins, the nuclear resident loses their visa, comes out to the cytoplasm, and binds to the 5' UTR and 3' UTR respectively to control the translation and the replication. And we showed that experimentally, and we actually now started believing that the nuclear protein comes out to the cytoplasm because of some modifications. So there is some post-translational modifications in these proteins, which kicks them out or sends them out to the cytoplasm. And with the differential affinity for the viral 5' UTR versus 3' UTR. Remember, the same protein binds to 5' UTR and 3' UTR. So it's possible highly that there is modifications involved, which helps them to recognize whether to bind 5' UTR or 3' UTR. So the affinity is altered due to modification. And obviously the availability or the abundance increases because of the nuclear to cytoplasmic relocalization. So, and that's the time Harsha Raheja, she stays close to Faridabad. So she herself exported to nucleus of IISc, and she showed that actually there are modifications. Mostly it's actually phosphorylations, documented in literature, that there are several phosphorylation sites in the HUR. And she demonstrated that if you mutate the S318A or Y200 to F or T118 to A, you can actually see that stop the migration of HUR from the nucleus to the cytoplasm. That means the phosphorylation is important for coming out from nucleus to the cytoplasm. And this is a very, very important event. If that is stopped, then the virus replication will also be stalled. So now among the three, she was actually perturbed that which one to focus on. And that's the time we took help from Tushar at the RCB. What we did, we just infected extract. We actually pulled down with the HUR antibody and did a phosphoproteomics, analyzed the phosphopeptides. And Tushar tells us that actually 318 position, the serine is actually important or more abundant when this HUR is actually relocalizing to the cytoplasm. So Harsha tried to look at it, that who is actually phosphorylating it and where it is. And eventually she showed that it is PKC delta, which actually phosphorylates this 308 positions of the protein, and that sends them out. So if you use an inhibitor of PKC delta, you can see there is an increase of the cytoplasmic abundance with the virus infection, which is called Japanese fulminant hepatitis C or JFH1. If you use the virus, there is cytoplasmic abundance increases. But if you use the inhibitor of PKC delta, you can see there is a decrease or it's coming back to the almost normal level. So you can stop the virus, stop the migration of the HUR protein from the nucleus to cytoplasm. Similarly, another protein I told you, PTB, that's also a nuclear resident that comes out to the cytoplasm. And there was in the literature showed that there is a serine 16, which phosphorylation is important. And serine 16, Harsha showed, is actually phosphorylated by PKA enzyme, another kinase. So essentially what we tried to show that all these proteins are nuclear resident, but eventually after virus infection, they come out after modifications, mostly phosphorylations. We identified the kinases that are phosphorylating them respectively, like PTB is done by PKA, HUR is phosphorylated by the PKC delta, and then they bind to the 5' UTR or the 3' UTR in a time-dependent manner. There is a time kinetics, there is a dynamics. I'm not getting into the detail of it, but we are actually working on it. And we try to show that how the nuclear cytoplasmic relocalization can regulate the virus replication. So it looks like inside the liver, life is not easy. It's a continuous tug of war between the host cell and the virus, and the post-translational modification actually gain the strength for the HUR, and for that, now they can tilt the rope towards the virus end, but host sends out MIR125B, which targets HUR and actually inhibits the HUR synthesis, and that's how it can keep it under check. But no matter how much you do, there is always OLA in the road. Its La protein actually changes it's the game changer. When the La joins with the HUR, it's the virus which wins. But eventually, everything is under the control of a traffic light. And these are the lights which we consider that decides the post-translational modification, that decides whether the protein will come out from the nucleus to the cytoplasm. And that's essentially we have been working. So we talked more on translation and now replication, and try to see from the proteins can we shift gear and look at some of the other characters. And that's the time RNA sequencing was getting popular. So we collaborated with, I think, SGPGI Lucknow, and tried to see whether we can actually have some serum samples from the hepatocellular carcinoma patient, cirrhosis patient, or a chronic hepatitis C patient, and do a sequencing of the RNA to look at which RNA is abundant there. And that's the time we found there is a long non-coding RNA called highly upregulated liver carcinoma, HULC. And this is the RNA that is abundant in the chronic hepatitis C and the cirrhosis case. And how it causes pathogenesis, we have published in 2019. We showed that actually the virus, after infection, the viral core protein goes into the nucleus. The core binds to RXRA, binds to the promoter of the HULC gene, increases the synthesis of the HULC mRNA. Now, the increased long non-coding RNA then actually helps in more and more lipid biosynthesis. ALK is known or shown to involve into the lipid biosynthesis. And this increased lipid droplets then actually helps in the HCV core protein loading into the lipid droplets. And finally, the viral RNA also gets loaded and uses the VLDL pathway to exit out of the system. So this is essentially, we solve the mystery of how a long non-coding RNA can help the virus exit from the cells through the VLDL pathway. But that is the pro-viral. But there is also another angle with the antiviral that Geetika shown in her thesis, that this Hulk also have a capability of sponging microRNA 122. What it does is actually sponges, and microRNA 122 is known to help the replication. So if it sponges out, it actually inhibits the hepatitis C replication. So Hulk in one way is actually helping the virus to egress out, and the other way it's actually reducing the replication. And actually in the tumor cells, it has been shown, there was a paper in nuclear in the Nature Medicine, that in actual hepatocellular carcinoma patient samples, in the locations of the tumor, there is always a lesser replication compared to that. So we started thinking there could be some factor, which we call oncogenic factor, which could be the microRNA 122 or one of these long non-coding RNA that we are trying to solve the mystery. So now with this long non-coding RNA, we extended it and tried to see, is it possible we can find out little more character to understand the pathogenesis that the hepatitis C causes. And there is Sachin Tripathi joined, and he said, sir, actually we see in the literature that in the paraspeckles are actually a lot of times making a change in the strategy for the pathogenesis for the viruses. So is it possible in the hepatitis C we can actually look into little closely at the paraspeckles? And the paraspeckles components are mostly the NEAT1, which is abundant long non-coding RNA, and along with there are three proteins, the NONO, the PSPC1, and the PSF. So we try to see, is it possible that we can see NEAT1 by RNA fish? And Sachin showed that actually hepatocytes, you don't detect or you don't see the NEAT1, but upon infection, you can see it started coming out and it started. So is that possible that the virus somehow increases the NEAT1 for its purposes? So we looked at it a little more closely with the tissue samples, and this we have done with the IPGMER Kolkata with Dr. Shoma Banerjee's lab with the HCC tissue samples, and she actually demonstrated that NEAT1 has two variant 1 and variant 2. Actually, they are upregulated during the HCC in the tissue samples, and in the serum samples also. Actually, it is more pronounced in case of the variant 2. We found that there is a strong correlations with the hepatocellular carcinoma and the upregulations of the NEAT1. So the obvious question, what will happen if you bring down the NEAT1? So what Sachin showed, that if you use SI NEAT1, there is a change in the NEAT1 level. And another thing that goes down, because there is literature that NEAT1 sponges mid-9 5P. So microRNA 9 is actually sequestered by the NEAT1. So if you actually silence NEAT1, you can see there is an increase of the mid-9 5P, as you see here. Now, mid-9 5P in turn actually targets BGH3, which is actually the target of TGF beta. And we are heading towards the molecule that directly correlated with the hepatocellular carcinoma. And it looks like this is the path that hepatitis C upregulates the NEAT1. NEAT1 sponges mid-9. Mid-9 cannot really stop the BGH3, which is central to the TGF beta. And the TGF beta goes ahead and then actually causes the pathogenesis. And this we tried to solve it using the tissue samples and also in the cell culture studies. Not getting into the details, I'll just try to give you a schematic that we tried to show. This is under normal conditions. The BGH is remains sequestered by the mid-9 or remain controlled by the mid-9. Now, under viral infection, the NEAT1 increases, NEAT1 comes out, it sequesters mid-9, so BGH3 free is now free to contribute to the progression of the hepatocellular carcinoma. This is Shochin's work. We are going to publish it soon. Another thing that we try to look at it is looking at the exosomes. Anji Pradhan, she's my international student, and she showed that if you really look at the microRNA in the exosome, often the exosomes plays a role in the transport of the viral particle from one organ to other organs. And for hepatitis C also showed that exosomal content actually varies with the disease progression. So she was interested to see, is it possible to analyze in depth the exosomal content at the level of microRNA, RNA, and at the level of protein. She did proteomics. She also did RNA-seq to find out which all microRNAs are up or down in the exosomes. And she zeroed down to one microRNA 375, and which she showed that in the intracellular level also it goes high, and exosomal fraction also it goes high in the infected cells. So the MIR 375 is known to goes high. Now, why it is important? MIR 375 actually contributes to the cell proliferation, and she demonstrated that actually if you use the BRDU assay or the migration assay or the MTT assay in all three assay system, or even the EMT assays with the vimentin and ecadrin, it suggests that this MIR 375 actually causes cell proliferation. If you use anti-MIR 375, you can actually reduce this cell proliferations. And actually the levels of the vimentin ecadrins also get altered if you use the anti-MIR 375 and somehow prevent the functions of MIR 375. So the obvious question is, now if you make the exosomes in the cells which are treated with MIR 375, what will happen? So she showed that if you use the exosomes from an infected cells, it causes cell proliferation. But if you treat the cells with MIR 375, infect them, and then so the exosomes now devoid of the MIR 375, that exosome, if you use it, it fails to proliferate to the same extent, suggesting, yes, MIR 375 in the exosomes, if it is loaded into the exosome, it contributes to the hepatocellular carcinoma progression. And this is again under revision and soon is going to be published. So all we wanted to show here that the HCV RNA upregulates one of the microRNA, and this MIR 375 goes loaded into the exosomes. And we also demonstrated that MIR 375 targets IgA BP4, and that's how it actually controls the hepatocellular carcinoma. This loaded exosomes then goes to the neighboring cells, and there it activates and it causes the EMT and the cell proliferation, and that's essentially the major root of the pathogenesis. While we were very happy doing all this molecular biology and cell biology, Dr. Vahan somehow impressed upon and said that you make a center for hepatitis C virus research, but go directly into the antiviral and then try some antivirals from herbals. And so I recruited one postdoc, and she started straight from the breakfast table, picked up the pomegranate and showed that pomegranate has all the mystery of compounds. And we isolated 2 compounds from pomegranate, which were actually, we were in the news for a while 5 years back, and again coming back into that because now we are trying to commercialize this. It looks like punicalagine and corylagine are the rich content of the pomegranate juice. And this actually, we saw the pathway, how it regulates the replication, how it inhibits the TGF beta pathway. And corylagine actually, we have shown that it somehow prevents the fibrillar collagen deposition, which is central for the hepatocellular carcinoma or the liver fibrosis. And we have tested these compounds in an animal model. Hepatitis C does not infect mice, but we have used a mice which harbors human hepatocytes. It's called a PXB mouse. It's in Japan. We collaborated with this company and got this antiviral tested. And we showed that in those, the compounds isolated from pomegranate and also a compound which is from Phyllanthus amaras, we could show that these compounds are inhibitory to the hepatitis C virus replication. Another thing that we try to show from the center is if we can make a vaccine candidate. As Pramod was saying that until today, we don't have hepatitis C vaccine when we have a range of vaccines available for SARS-CoV-2. It's largely difficult. It was very difficult because we didn't have a proper cell culture assays. We could not really make the virus enough amount. We could not test our hypothesis mostly. So when the cell culture available, then we tried to make a VLP, which is a virus-like particle, but which lacks the genomic content. So it doesn't have the genome, so it is non-infectious. And this VLP model, we actually made it from genotype 3A, which is actually prevalent in India. And we showed in a mice model that you can make this immunized mice sera can effectively neutralize the virus. And now this VLP vaccine, we are testing it in pig. Pig is just for the immunogenicity, not in an infection model, but we are just collaborating with another laboratory called Shipra Laboratory in Hyderabad, where the preclinical trials we are aiming to start soon after the pig results are actually in place. And in parallel, we are also trying to add a DNA vaccine along with the VLP vaccine to increase the cell-mediated immunity, which we can't really get that much amount when we use a VLP-based vaccine. That is good for humoral immunity, but that's also in the card, and we have been trying to it. So as I just told you, the path, we started with simple translation and go on to a translational mode, which is central to THSTI. So I show you here that we started with a small RNA SL3RF, ERF, which is patented, which inhibits translation by binding to the ribosomal S5. We also made a peptide from the RNA binding domain of La protein, which is a major player for hepatitis C translation and replication. We showed this peptide 7-mer, only 7-mer peptide. If you just add 6 arginine residues, with the 6x arginine, it can go on its own inside the cells and kills the virus. We have demonstrated that, we patented it. We have also shown that if you make a peptide corresponding to the RNA binding domain of NS3 protease, you can block the virus replication. In parallel, I just told you that phyllanthine corillagene and polycollagene, these are the compounds that we are direct antivirals. We are trying to stop the replications using this. We also collaborate heavily with Dr. Anjali Karande's lab, where we have isolated some monoclonal antibodies based on the neutralizing antibody from patient sera who have effectively cleared the virus, and isolated those. And based on that, we made monoclonal antibodies, and these monoclonal antibodies were able to block the virus entry effectively. And all these things were tested in the PXP mice, which are very, very expensive, but was only possible because of generous help of our beloved Professor N.K. Bhan and the Hepatitis C Center of Excellence. So now, finally, as I told you, being at IISc, we always explore new things. And one of my new thing is something where I need heavily input from Pramod, is we are trying to understand the mechanobiology of the liver. Now, as you all know, that liver disease progresses from healthy to fibrosis to cirrhosis to cancer. And, hepatitis C, since it goes through these same steps, we are trying to understand, is it possible that we can understand the role of the stellate cells and the hepatocytes and the ECM, the extracellular matrix protein, and how they accumulate and then the causes. So what we are actually falling into a model system where we try to simulate a situation where we use different concentrations of acrylamide. We make a hard gel with 40 kilopascal, or intermediate gel with 10 kilopascal, or a soft gel with a 3 kilopascal acrylamide and bisacrylamide cross-link. And we grow the cells in those. The idea is, now if you grow the cells in the soft or intermediate or hard gel, you can actually simulate or mimic the disease and the treatment in this condition. And we try to reverse the situation and see, is it possible to have a regression of the fibrosis in this particular model? And what we measure is the stiffness of the cells when they're grown in a soft gel or an intermediate gel or a hard gel, and try to see whether we can correlate the stiffness with the hepatocyte function, means whether now the hepatocytes functions are impaired because of the corresponding stiffness. And eventually we would like to see see, the moment you say biomarker, people will jump on. So we really don't want to say that we are looking for a biomarker, but we're trying to see whether if you can have a biochemical marker, which will help to add on to the existing potential clinical therapy along with that. So fibroscan, then serum marker, which is currently available, can we really add on to that and have an improved assessment of the fibrosis by using some biochemical marker, which is actually, we hope that we'll get a lead from the mechanobiology, and we'll be able to add on to the treatment options. So what we essentially do, we take the normal liver, fibrotic liver, and cirrhotic liver stiffness model using different, as I told you, 3 to 6 kilopascal, 8 to 12, or 20 kilopascal fibroscan measurements are like that. And these polyacrylamides are actually measured, the stiffness are measured by the AFM. I have a multidisciplinary student. We have a bioscience and engineering department, ESSC, and in this particular department, which we used to call bioengineering. And we have a student from mechanical engineering background and working on the biology problem. So I have a collaboration with Dr. Ananta Suresh, who is actually a professor in the mechanical engineering. So we try to see, is that possible that we use two different cell types? One is the HUH7, which is hepatocyte-derived carcinoma cell line, but obvious choice is definitely primary hepatocytes, but we don't have the access. We are looking for the access. And the LX2, which is immortalized hepatic stellate cell line. And then try to establish an assay system to have a substrate stiffness and the characteristics of the liver cells in the context of fibrosis progression, and find a correlation, if it exists at all, between a stiffness and the hepatocyte function. And to understand the molecular mechanism of the hepatocytes involved, and in particularly in a fibrosis progression. Why we are interested? Because we think that the hepatitis C viral proteins, particularly the core protein was known, but now we are looking for the non-structural protein also, how it can actually contribute to the disease progression. Can we test this hypothesis in this particular model system? So we are using cell types called replicon cell line, which harbors the non-structural protein of hepatitis C. And we use the HUH7 cells, which is control, and try to compare their stiffness by growing in different acrylamide concentrations, and then try to see, is it possible to make some headway from or some early leads. I just mentioned that hepatitis C was actually running low for a while, but just came into, again, limelight 2020, when these three brilliant scientists, Harvey Alter, Michael Houghton, and Charles Rice, were given Nobel Prize for the discovery of hepatitis C virus in the physiology and the medicine. So, but hepatitis C, still it is not solved, as Pramod rightly mentioned in the beginning. There are a lot of issues which are unresolved yet. Number one is the appropriate model organism. We can't use chimpanzee, it's now banned. Mice, hepatitis C does not infect. Humanized mice or ectopic liver generation, people are using the surrogate models, but it's very difficult, very, very expensive experiments. Vaccines, there are leads, but nothing has consolidated yet. Variations in the viral sequencing, error-prone replication by NS5P is a major hurdle. Then extensive glycosylations of the viral envelope proteins, limited production of the whole inactivated virus, so we can't really inactivate by making tons of virus. Even if we are growing in the cell culture, it doesn't grow to the extent desirable in a fermenter. Another thing is, a lot of patients, even after the virus is cleared, they progresses to hepatocellular carcinoma. That means some of the pathways are activated, and unless we take care of those pathways, cannot bring down those pathways, it's difficult to stop the progression of the disease. So the activated pathways, the epigenetic modulations that happens in a patient, which actually varies from patient to patient. Not all the patients are actually so unfortunate situation, but some patients, very unfortunately, they clear the virus, but somehow some changes happens. We're trying to look at those epigenetic modulations or triggering the secretory molecules. So this ECMs we talked about, we need to see more in detail to find out the answer and stop the progression to hepatocellular carcinoma. So soon after Charlie got this Nobel Prize, I was in touch with him since my postdoc days in UCLA. So Charlie said that let's hope that this example of the benefit that science can bring will encourage the public to embrace and support fundamental, curiosity-driven research. As we are in the midst of experiencing with an ongoing pandemic, there are many challenges ahead. So essentially, I take the lead from that, and I feel that hepatitis C is just a system. It can be extended to other hepatitis viruses, but it's very important that we have a system in place and we have a network in place and we work together. As I'll just now show, my network for hepatitis C virus research are such a huge group of people that I collaborate. For the antiviral group, these are the collaborators. Unfortunately, we lost our beloved friend, Professor N. Srinivasan from the Molecular Biophysics Unit at the Indian Institute of Science. We collaborated mostly on the computational and the structure-driven analysis of different molecules. Then N. Jayaraman in organic chemistry, Siddhartha Roy in the Bose Institute, Apurva Sahu NII in Sanjay Dutt. And then host-virus interaction, we have a pathogenesis group where I'm actively collaborating with Dr. Shiv Sharin's group. We had a project together. Rakesh Agarwal's group when he was in SGPGI. Now I'm still continuing with his other disciples at SGPGI. Ravi Kumar and Shoma Banerjee in PGMER in Kolkata. As I told you, liver mechanobiology is again my luxury project where I try to understand something which I don't understand. Cell stiffness, nuclear shape, and laminopathy. We published some on these areas with Anand Suresh, Dr. Naren Dikshit, and Roop Malik and TIFR. We work actively on the liver-targeted drug delivery because see the molecules that we have made, we have to make sure that they are targeted to the liver. And for that, we have demonstrated that we can make use of virosome. We took help from Debbie Sarkar at ISAT, in Delhi University South Campus. We have taken help from Dendrimer with N. Jayaraman, Ajoy Sood and Prabal Maiti. They are physicists. It's a wonderful experience working with the physicists. And nanocapsules, this is with the materials engineering. We use as different nanocapsules. GLP and DNA vaccines, actually it started with Indo-Australian project, but now we are doing it ourselves. And monoclonal antibody and neutralizing antibody, as I told you, we are working with Professor Anjali Karande's lab, and later on we work with Professor M.S. Shaila and Dr. Soma Das. And we have been working on this to see how we can somehow make it translational, because all this work have very, very interesting leads. And that is only possible because of this wonderful group that I have it for last 23 years. As you see, this is my current group. And there are 3 postdocs I left behind at NIBMG. They are continuing there, Trinath, Rajiv, and Anindita, and rest of the people are with me. Some of them are already logged in. The work that I have shown here is, I can't see Anji here, Anji, Harsha, and Sachin Tripathi here. And the rest of the people are working in different areas. The lab is big, and we have different other projects. I didn't want to dilute it and wanted to concentrate only on hepatitis C. And it's a real wonderful journey. And we hope that at the end of the road, we'll find out some translational product and which will help to the society. And that will be our contribution to the society and the human health. Thank you all. Namaskar.
Thanks a lot, Soumitra, for an excellent lecture. I mean, mesmerizing the way you have conducted all these very fine and complex experiments over last 20 years or so. And you just told it like a story. Fascinating. And, this is so important. And I was, intrigued and very impressed by that message that you gave, that the viral RNA there is 2 competing things. One is the replication and the other is protein translation. They are so fundamental and very, very impressive. And many people think that, these fundamental biological science may not truly translate, but But everybody understands that the Nobel Prize was given for fundamental science, not for the discovery of the drug, sofosbuvir. So it is the fundamental science which actually resulted in the drug molecule that is effective. And Harvey Alter, in I think early 90s, he wrote an editorial, Descartes before the horse. I clone, therefore I am. That was the title of that editorial published in Annals of Internal Medicine. So they were the ones who actually cloned the virus, showed the sequence, and the virus wasn't visualized till then. So it was among the first viruses which was recognized, given a name, although the virus was not seen till that time. So obviously, I think this is very, very important science. And the other thing I was very happy is the stiffness that you are now testing. So when liver becomes cirrhotic, one of the problems that we see clinically is development of portal hypertension. And that portal hypertension is primarily because of the fibrosis and distortion of the architecture and stiffness. So that leads to clinical consequences of, development of varices and GI bleeding and many other complications. But in addition to that, whether stiffness by itself causes hepatocyte dysfunction, I think this is an important area that you are engaged in. And I think this will also result in some clinical benefits if you show some biomarkers or some targets. So that's what my comments are. And hepatocyte stellate cell is something I think also one of the major areas, and I'll talk to you separately later whether something can be done on that. So over to Milan and Ramandeep for questions and answers. I'll stop there.
Thank you, Professor Garg.
Now, the other thing I want to say that, because of this fundamental science that Soumitra Das is a fellow of all three national societies and also a JCBOS fellow. People think that you get these fellowships just like that. No, sorry. I mean, you have to have hard work going on for years together before you actually are recognized by your peers and your seniors. So congratulations, Soumitra, for that. And
thank you.
Milan and Ramandeep.
Be happy to collaborate, Pramod. We'll talk more on the stiffness. Yeah.
Sure.
Thank you so much, Professor Das and Professor Garg, for this insight. And it was really an honor and pleasure to listen to your talk. So the talk is open for questions. We will request everybody to post their questions in the chat box. Some of the co-hosts can ask the question directly. Dr. Ramandeep had some questions, so I would request him. Sapna, kindly unmute Dr. Raman so that he can ask his question.
Done.
Dr. Das, nice talk. I had a couple of questions. Do you know what are the conditions which regulate the levels of HELC non-coding RNA in the cell?
See, what are the conditions means like other than viral infection?
Other than viral infection? Or is it only viral infection which regulates?
Right. So, other than viral infection, we haven't really explored much. But I think DNA damage conditions, there are reports that it actually increases some of the long non-coding RNA, and HELC is one of them. But I haven't seen much of those work on this. Any stress may not really regulate. I mean, unless it is a liver specific.
So do we know which is the viral factor which controls these levels?
So our study suggested it's the core protein of hepatitis C. If you overexpress that, then it actually goes high.
Okay. And my second question was that are there any known modulators for microRNA 5P, I think it was, at BGH3, known which can be used as maybe antivirals? Is it like
right? That's basically Sachin's dream is. Sachin can answer that question, but only thing is we haven't got into the antiviral mode for this. This is a new findings. We're trying to publish that and to show that how this can be targeted. If this MIR9 and this, it's very difficult to show the microRNA and long non-coding RNA interaction. So we're actually struggling to show physical interaction between to prove the sponging effect. We have all these indirect roles. We are trying to do trap assay to show that MIR9 is actually sequestered by the NEAT1. So once that is done, then we'll try to see how that what happens to the BGH if you just upregulate one of them. But certainly, yes, if this target is genuine, then it can be a good target for exploiting for antivirus.
And it seems like there's a lack of a dependable animal model in the country, right, for hepatitis C virus. So are there any efforts somewhere?
There were actually, see, at one point from DBT, we have taken an initiative to make some kind of a mice model for hepatitis research. And that time there was a call for proposal and several projects were funded. I think if I'm not wrong, 7 to 10 projects were funded, but everybody ended up publishing one paper and left there only. So nothing translational happened, but I think I was just talking some other day with Pramod. This is something which is very important for hepatitis research in the country. And being a gastro person, I think he might be interested to push it through in DBT and see whether we can revisit this and have a central, rather than giving money to 7 different places. If we can have a centrally somewhere funded and everybody put their effort to make it's not a big deal. People have done ectopic liver, publishes, and there it ends. So the leads that we have got from different places, we also showed that if you see mice cannot really replicate hepatitis C. Then we found that the mice does not have the LA protein. They have the LA protein, but that has some mutations in it. But if you knock out and knock in the humanized LA, then that mice can be actually able to promote the replication of hepatitis C. So that is again a wonderful leads. But as Pramod said, I'm a basic research scientist. So the translation doesn't really happen in this place. People try to publish and then submit their thesis and go. So it becomes difficult. But those leads can be consolidated in India nationwide and try to have a, definitely it's important for hepatitis E research, hepatitis C research. The other day, Pramod was talking about hepatitis E research. Again, there's the same problem. I think we don't have any animal model proper.
Yeah.
It would be good to have it.
Yeah, thank you. I will request now Milan to ask you a few questions. Yeah.
So there is another question from Dr. Neeraj. Neeraj, if you can ask your question.
Thanks, Milan. Thank you, sir. Wonderful talk, actually. And it was nice to see the research, basic research going to the translational side. So I have a couple of questions. So, but in the interest of time, I will ask only 3. So, sir, my interest was like, you said that post-translation modifications were regulating the affinity of protein, translocation of protein. So did you get any idea like what signals? You said there may be some signals. So did you able to zero down which signal it was?
Right. So basically, we zeroed down into the kinases, and we found that there are kinases and the proteins also, you know, it's a dynamic equilibrium. They just post-translational modification happens and then they goes back, again comes back. So other than kinase, we haven't really addressed the other protein modifications. But, you know, if somebody takes up as a project for extensive proteomics, I think that would be a wonderful way to look at it, because we have the targets already identified, but we really don't know what all modifications is possible. But other possibility I can think about is glycosylation. That can affect the binding, particularly protein-protein interactions. So we haven't really explored, but kinases, since there are phosphorylates and then kinases inhibitors are available. So we have actually gone for the inhibitors for the kinases.
Okay, that's good. And sir, my second question is, you said that some microRNAs were selectively packed in the exosomes and then secreted, and they were having impact on their migration, invasion, proliferation, and all those things. So you said microRNA 375 was the key the lead identified from this. Did you get chance to have the look on the expression level of this microRNA 375 in tissue, as well as some serum or from the patient or something?
See, microRNA 375, Anji, are you there? I don't think we have checked it in the tissues, but I think we have tried in serum, and that results I have shown you that it is upregulated there. In the serum
you have tested and tissue. No, okay. And sir, the last one is like you have shown multiple leads that were translational, that had the translational opportunity. So I know all are very important for you, but do you feel like there is one thing that you think is good enough to go to the market and has more potential than the another one?
No, so strictly speaking, after the drugs are already in the shelf available, so more interest is on the vaccine development.
Okay.
Because if you can prevent it, nothing like that. So VLP vaccine, now I am more concentrated into this and try to see, because we have also a VLP vaccine for SARS-CoV-2. So both the projects are funded by DBT, and we are actually going in tandem and trying to see whether we can go into the second level for the preclinical trial. But the major problem that we are facing for hepatitis C is we don't have any animal model. For a challenge model, now we are trying to talk to MOD, vaccine center. Try to see in Rafi is there. So we are trying to see whether Amara, he was a graduate student when I just joined as a faculty here. So Rama was saying that they have some monkey model to test vaccines. So I'm trying to see whether that can be explored and see whether we can test this vaccine candidate in that particular system.
Thank you, sir. Thank you very much, sir. Over to you, Milan.
So next question is from Dr. Susmita. Susmita, can you? Susmita, can we?
Yes. Hello, sir. Thank you for a wonderful talk. I wanted to ask, I mean, we know that hepatitis C mostly shows virological failure because, you know, there's a quasi-species concept. So the complex formation, which you showed, could be a very good target for drug discovery. Is it variable if there is an NS3 mutation or an NS5A variation? Have you seen any of that?
So you mean the replication complex, whether that
Yes.
will vary or not?
Yes. Yes.
We haven't tested, but I don't think that NS5B or any one of those non-structural protein mutations will have changes in the replication efficiency as much. Well, we haven't tested, but one way to do it, if there is a mutation that changes the conformation of the protein so that it does not really crosstalk with the other players like HUR or LA,
Right.
then there is a possibility.
Right.
But we are actually thinking in another way, which I didn't really show. One of my postdoc, Ashutosh Shukla, he has been showing that P53 also somehow binds to the UTR, 3' UTR, and it's actually changes the replication complex. So that we are trying to prove it in the detergent-resistant membrane fraction isolated, and then try to show that how the stoichiometry is changed because of a nuclear induction into the complex. That is possible, but mutational as such, we haven't really seen it. See, this mutation things is an eye-opener after the SARS-CoV-2. Now everybody started looking for variant mutations. So we'll have to look at it again. Yes, thank you.
So the next question is from Dr. Chris. Chris, if you could ask your question yourself.
Hello, sir. Can you hear me?
Yeah.
Yeah, we can.
That was a wonderful piece of work as well as the talk. It's amazing that how much you could accomplish within last 2 decades. So one question was, it's pretty intriguing to me why mice are so, immune to this HCV. Is it possible that understanding how mice are immune to HCV would help to identify specific targets that could help you to control the HCV infection in humans?
You said it. See, essentially, it's the host factor. We believe that there could be some host factor requirement, which is not, it's not that the virus cannot enter. So people have tried, Charlie Dice have shown that if you have a transgenic mice, which actually express the receptors, occludin and claudin, then that virus can actually enter the mice. But that's it. But it can't really replicate efficiently because the host factors are missing. One of the host factors I told you that we showed that if we use humanized LA, and we are trying to do a CRISPR, but I have to look for a person who can do that. So if you do a CRISPR with the humanized LA, and then we can actually address. And under that milieu, if you do an RNA-seq and try to see, as you said, that what goes wrong because of the absence of something, what other things that are maybe the other players have, all the stoichiometry is disturbed. That needs to be restored to do that.
Thank you, sir.
So next question is from Dr. Bhavtosh Das. Bhavtosh, can you directly ask your question?
Good afternoon, sir. Can you hear me?
Yeah, Bhavtosh, tell me.
So, sir, since you are directly involved with this COVID-19 research, so just I was wondering to know, do you find any correlations with the hepatitis C infection and the severity of COVID-19 in the patients?
No, see, hepatitis C is a chronic disease, and this question should be targeted to promote only, because what happens in the liver, whether the onsets are, but I believe the effect of SARS-CoV-2 on the liver, if at all, in a long-standing mode, will be only known after a few years, because hepatitis C takes 12 to 17 years to show up. So I don't know. Pramod, you might be able to answer it.
So if the question is that what does it do to the liver, I mean, there are now ample literature that it causes multiple problems. One, that it can induce hepatitis and your enzymes may go up. That's quite common actually, but mostly, subtle and doesn't cause major problem. In patients who have underlying liver disease, for example, due to hepatitis C virus or B or alcohol, their liver dysfunction may worsen. And some of these patients go into liver failure. So these two things are now well known. Whether in people who have normal liver, long-standing long COVID will result in some dysfunction, we don't know. As you said, that it may take a long time, but these two things are very well known now.
Yeah.
Thank you, sir.
So next question is from Dr. Guru. Guru, you could ask your question, please.
Hello, sir.
Hi, Guru. How are you? Good to see you.
Nice talk. I wonder how you keep adding so much data every time.
Don't.
Sir, my question is generally, actually, it's a generic one. So it takes 15, sometimes 20 years for people to really, get severe liver cirrhosis, right? So I believe the virus does this by really replicating at a very low level and evading the immune response. So is there any effort in terms of, monitoring patients over a long time and see what are the factors that triggers? Why does the virus suddenly, start replicating so that it causes damage to the liver? Maybe Professor Garg also may have seen incidences where suddenly somebody starts becoming severe, but they were diagnosed much earlier. Is it really like some factors that triggers this, not related to virus directly?
See, one way to think about this, if the virus keep on replicating, but can still mutate and makes one quasi-species which is much more severe compared to the one that has entered. That's a possibility. Other possibility is actually slow damage and reprogramming of the liver system. The whole microenvironment is changed over the years, and that might somehow, and that depends on the individual to individual. I don't think it's a very generalized situation, right? Pramod, what do you think?
So there are a couple of situations. One is that most people would continue to have very, very slow damage, and that's the reason in hepatitis C, it takes about 20 years for someone to develop cirrhosis. Now, even after they develop cirrhosis, I mean, even if you have only 10% liver functioning, you can have almost a normal life, a compensated liver disease. And that is why many people die with cirrhosis rather than of cirrhosis. That's something that clinically we know. Now, what happens in some people that suppose somebody is immunosuppressed because of some other reason, he is on immunosuppressive therapy and then becomes immunocompetent again. That is the time there could be a flare. This has been very well shown in people who have got hepatitis B virus infection. In C also, someone who develops say any other cancer is on chemotherapy will be immunosuppressed. And once the chemotherapy is stopped, the immune system rebounds and then they have a flare in the acute hepatic injury. But more important clinically and more common is someone with chronic hepatitis C and liver disease may develop another insult. For example, they have hepatitis C virus infection, which is quite common in this country, or they have drug-induced liver injury. So you have a baseline slow liver injury going on, and then you have an acute insult like this, and your liver functions drop like this. and they develop what we call as acute and chronic liver failure. So that is quite common. In fact, it has a very high mortality in such people.
Thank you, sir.
Yeah.
So there is a question from Dr. Pallavi. Pallavi, if you could ask your question.
Hello, sir. Very nice talk. And I was more keen towards...
Pallavi, thanks for your help to measure the exosomes. Tracker.
Yeah, so I had a question regarding the exosomes only because I'm more interested into that. So you have shown that the exosomes get secreted and then they go for a paracrine type of signaling where they increase the proliferation. So have you, first of all, tried doing any EM images to show that it exactly happens, one? And second is, what type of cells do they interact with? Do you think that would be something which could be done?
So you mean to say that it comes out from the liver and then goes to other organs?
Yeah, means I just need to see what are the types of cells that they are communicating with. Is it something that would be?
We haven't seen, that's a good question. We haven't seen it. So you are trying to say that these exosomes that are isolated, you are only adding to the liver cells. How about that adding to, let's say, some other neuronal cells or some cardiac cells or something, and see whether that enters there or not?
Yes.
See, one thing you have to keep it in remember, in this case, it's not receptor mediated endocytosis, right?
Okay.
So it's very likely that it will probably get in. So, and that's basically the whole hypothesis behind exosome is that gives the flexibility from organ to organ transmission, right? So, but we haven't tested it. We can easily test it. It's not a problem. And EM image, we don't have that much of accessibility and expertise here to look at it. But if you would like to do it, we can always collaborate with you and then do a little more on the exosome characterizations, which I recently wrote to you. Now we wanted to get into more on this hepatitis C and also in the dengue and see how we can do better on this characterization. Definitely their shape and sizes are changing with the content.
Okay, great. Thank you, sir.
Thank you.
Yes, so Dr. Das, I have a general question. Rather, I request you to give your opinion. So the question is, how did these iris elements evolve and why only certain pathogens have it? Because you have so many years of experience with iris.
That's a good question. See, the only thing is I can see this is evolved in the RNA viruses and RNA positive strand RNA virus. This is an initial obligatory step to have a translation going on. If they don't have CAP, and particularly those who shuts down the host cell protein synthesis, then they have to find out a backup mechanism. Well, it was discovered in polio, but I don't know why hepatitis C is having it, because hepatitis C does not really completely shut down the host cell protein synthesis, although it is impaired. But they probably evolved it because they will have less competitions with the host. Because remember, they are having 40S binding straight to the UTR without having any initiation factor. So the requirement or dependency on the host is limited or restricted. And that's how probably the evolution happens.
Right. Thank you. So since there are not many questions, I would request Dr. Susmita to formally thank. Susmita, if you can take over.
Hello, sir. Wonderful talk and really a nice time. We were expecting that we would, invite you to the campus to be in person here so that we can have an elaborate discussion after that. But we'll do it very soon. And thank you so much for your time. And thanks to all of you for, patiently hearing and taking part in the conversation. Over to you, Milan.
Where is his memento, bhai?
We are saving it. We are saving it.
No, that we will be sending. We are
saving it. We'll be sending it to you.
Yes.
Sure. Thank you. It's always a pleasure to interact with all the friends at THSTI.
Thank you.
Any of you,
we are looking for collaboration
when you come to Bangalore.
Sure.
Please visit IISc.
Yeah.
Pramod, keep it in your cart.
Sure. Thank you very much. Bye-bye.
Bye. Thank you.
Bye.
Thank you.